Hello, in this video, this is a continuation of the last video on, sorry, that seems a little weird whenever I stand, the basic concepts of metabolism. This is the second video to that, okay? So let's talk about it. We're going to be continuing our discussion from intracellular transduction. So intracellular transduction is also called intracellular messenger system, okay? Which is pretty much just cell signaling, right? I would highly recommend, please watch the previous video if you haven't. But anywho, let's get into it. So there are two types of intracellular messenger system. One is adenylate cyclase. Oh, by the way, disclaimer. This is a video for self-revision purposes only. This is for me, okay? Me speaking to my future self in case I forget, during the finals. So, I don't just like freak out. There is me explaining stuff. Yeah, so we don't cram. Okay, so there are two types. One is adenylate cyclase system. It took me a while to pronounce that. And another is calcium or phosphatidylinositol system. Okay, so let's talk about this one. Hold on. Okay, so right now we're going to talk about adenylate cyclase system. Okay, so this one, how do we recognize or differentiate it from the other one, which is calcium or phosphatidylinositol system? Okay, so how do we differentiate this? First of all, if the tissues, it responds to more than one sort of chemical signals, it has a lot of receptors. Like, let's just say one cell membrane has lots of receptors, or a lot of receptors. That means it is linked to adenylate cyclase system. Please, I do not want to pronounce this again, okay? This is characterized by a few things. These receptors are characterized by a few things, which is ligand binding region, 7 transmembrane helices. I've got no idea what that is. An intracellular domain that interacts with G proteins. Okay? Wow, that was crazy. Okay, so we're going to talk about it step by step. So imagine that there's a protein, there's a receptor, I think. There's something, okay? There's a CAM-dependent protein. Yeah, I'm just reading stuff out, okay? Protein kinase. Okay, so what happens is it's made up of, let's just say, 2 units. One is a regulatory subunit, the other is a catalytic subunit. Okay, so what does adenylate cyclase mainly do? It converts, it helps in the conversion of CAMP, which is cyclic adenosine monophosphate, from ATP. Let me just write it down. ATP to CAMP. I'm gonna refer to this as CAMP because it's easier, which is done by this, adenylate cyclase. Okay? So this is what it does. Now here's the thing. There is, like I said, a CAM-dependent protein kinase, which has 2 subunits. Okay? This one is regulatory subunit, and this is catalytic subunit. Sorry, I've been writing all morning. That's why it's getting kind of fuzzy, right? So what really happens here? We're gonna get into this a little later. No, we're gonna get into it right now. So this breaks into like this and this. This and this. What causes this breaking? This gentleman right here does this. Like it causes it, okay? It doesn't really break it, it causes it. So this catalytic subunit, okay, then ends up, so this is a catalytic subunit. That means it is sort of like a catalyst. That's what I like to think, okay? So there is, let's just say, a protein substrate, okay? A protein substrate. It wants to get along with inorganic phosphate. So this thing kind of like catalyzes this thing. And whatever happens, let's just say this, which is a phosphorylated protein, this in turn causes the intracellular effects. As in, this is the one which actually makes changes in the DNA, right? And then it later gets dephosphorylated by protein phosphatase. Get it? I also kind of don't. Okay. Deep breath. Hi. Okay, I figured it out. Okay, so let's talk about G proteins. Okay, so what is the function of G proteins in the adenylase cyclase system? Okay, so what G proteins do is that it forms a communication link between ATP and adenylate cyclase, so it can produce CAMP. We kind of established, I'm going to call it CAMP, right? CAMP. So, yeah, this is what it does. So then we have this thing. So this is a thing. Let's just say it's an enzyme that is still not activated, okay? So this enzyme is catalytic subunit and regulatory subunit. So in the presence of regulatory subunit, you cannot really use this one, which is the thing that we need to use for, catalyst and all. So this CAMP, it kind of helps in the breaking down between the regulatory subunit and catalytic subunit, okay? Oh, sorry, I forgot to draw one little thing. Okay. So then, what this does, okay, is that it participates in this reaction. Okay, so let's talk about what's happening here. So there is a protein substrate, right? We have a protein. Yeah, we have a protein. Okay, the protein wants to form bonds with the inorganic phosphate, which is coming from ATP by conversion of ADP, right? So there is protein kinase and the catalytic subunit that we just separated from regulatory subunit. These two work together to make it a phosphorylated protein, okay? This is the thing that works for the intracellular effects. Okay, this is the one which, kind of binds into the DNA and stuff to like do your thing. Do, whatever thing that you want. Maybe, you want to get tanned, vitamin D, or like, your retinoic acid, all that stuff that we talked about, the intracellular receptors, right? So this is the one which really works on your thing. And then, so like imagine you're going to, those spray tan places. I don't know how it works, but I'm guessing they just do something with the receptors. Let's just imagine that, okay? So you wouldn't want to get tanned up, like you want to get tanned up to a point, okay? Like we all want to get tan. Like I think I like my skin a little dusty, right? I don't like it too tan. I like it a little dusty, right? But at the same time, if I get like a lot tan, I look very orange, right? Which I kind of don't like, but you know, I like dusty on me, right? So here's the thing. I don't want to get as much as tan, right? So doesn't our, like, our body, of course, doesn't want it to like just go on and like to just like happen. I'm sorry, oh my god, English is not Englishing. Okay, bear with me, all right? So now what really happens is that we have another thing right here, which is protein phosphatase. Protein phosphatase. I don't think yeah, we can see it. Protein phosphatase. So this thing actually dephosphorylates the substrate protein and forms a dephosphorylated protein, and it separates. Okay, I don't think we had to write this. It separates the, not this as well, it should be here. Okay, it separates the phosphate, and it also gives off water molecule. That means it's sort of like a condensation reaction, I believe, maybe. I'm not sure. Okay, so this thing in turn reacts with GAMP and hydrolyzes it, so it does not, it stops initiating this. Get it? It's a good example of maybe that kind of inhibition, like product inhibition, maybe. I'm not sure. Okay, forgot to say this, but it converts CAMP to 5' AMP. 5' AMP. Okay, so next up we have calcium slash phosphatidylinosytol system, right? So stay tuned for that. So this one is mainly, it uses activated phospholipase C, okay? So it's like, let me explain. Okay, so this is calcium and phosphatidylinosytol system. Okay, so what really happens is this is your cell membrane and this is your phospholipase C. Okay, it's like a receptor. It is a receptor. Okay, so you have your thing binded right here, the hormone. Okay, when the hormone binds here, this thing, it becomes activated phospholipase C. And then, okay, I might be scientifically wrong for this, but just bear with me for the explanation, okay? This thing forms into two molecules. One of the products being inositol 1,4,5 triphosphate. The other being diacylglycerol. Diacylglycerol. Yeah, okay. So both of these are second messengers. Okay, so what really happens is this one binds to the endoplasmic reticulum and starts spewing out calcium ions, and this one hold on. Okay, this one then does the formation of protein kinase C, which in turn participates in our last adenylate cyclase system in the protein kinase thing, and then the intracellular systems. And this one, since it produces calcium, we need another calcium ion, right? This one then binds to calmodulin. Okay, and it forms a complex, okay, which sort of activates enzymes to do the things. Sorry, the last part is a little hectic. Anywho, I hope you understood stuff from this video, and I will be posting more, hopefully a little less confused next time. Please watch my videos. If I made any mistakes, please comment down below, and I don't know, comment, okay? I like negative comments as well. Bye.